I'm not touching any of it. Okay. Image. I'd like to start by doing something that may not sound like science, but scientists ought to be responsible citizens as well. So I'm going to give a couple of minutes to people from the 1,000 Hour show.
How's everybody doing today? Good. Staying warm, I hope. It's actually, I know it's actually but anyway, well, let's start off with a question here. How many would like to go to a free rock concert? Totally free. No charge. Yes. Well, my name is Molly, and I'm going to tell you guys all how you can do that. I'm part of the 10,000 Hour Show. Has anybody heard of the 10,000 Hour Show there, hopefully? Awesome. Well, 10,000 Hour Show is basically a student organization here on campus that engages young people in the community and celebrates at the end of the year with a rock concert.
For those of you that haven't heard of the 10,000 Hour Show, it's really simple and it's a lot of fun. I started doing it last year, my freshman year. I just served as a normal volunteer. I did my hours, got to see OK Go. It was a really fun concert, really good time. This year I'm doing grassroots, and a lot of you might wonder where you go to get started with 10,000 Hour Show. We just launched a brand new website. It's really cool. Check it out. It's www.10000hoursei.org. You log onto our website, get registered, pick some organization, do 10 hours, and we'll give you a free ticket to a rock concert. A lot of you might not know where you can go volunteer, what you want to get involved with. If you log onto our website, we have a list of over 200 nonprofit organizations from Johnson County. You can pick whatever fits your interests. If you're already doing volunteer hours, you can register through that, and that can count for your hours. Or our organization holds V-meets twice a month, and you can get involved with those and go with a big group of us.
Yeah, we've actually had Guster, Kate, Ben Folds, OK Go, my personal favorite ever last year. It was really awesome. And this year, in April, we actually don't know the band yet, but in a few weeks here we will know. So keep checking back with our website and make sure you do that and see what band we're going to have this year. It's going to be in the IMU Ballroom. And actually, we also, like we mentioned, it's 10 hours to get a free concert ticket. But we're starting the volunteer incentive program this year. And if you do more than 10 hours, say you do 15, 20, even up to 100, you get more, like water bottles, t-shirts, even for 100 hours, a free limo to ride to the concert. So get all your friends to come. We actually have a volunteer fair at the IMU this Wednesday, the 28th, from 11 to 3. So you can all come check it out. There'll be non-profit groups, and actually 10,000 Hour Show will be there too to help you guys set up if you'd like to be interested. So thank you all for your time. Thank you, Professor Fulton, and enjoy class, guys.
Do you want me? I can have them put them up. Okay?
Thanks very much.
Sure. Great. I have a special fondness for 10,000 Hours because it was a program that was started under my service as director. So, yeah. All right. There is a timer running on this. So on Friday we went over general course organization issues and started to discuss Chapter 1. Do people have any questions either about the course itself or about the material from Chapter 1 that we discussed on Friday? Okay, we'll keep working on that. Folks are doing pretty good at moving into the center, but we need to make sure we keep that up. It's a tough room to work in. So we mentioned the polysaccharides, and we're ready to move on to some very general issues of what sometimes is called physical chemistry. Physical chemistry kind of underlies biochemistry. We're all, at least, incorporate physical things, and it drives us too. And we need to sort of refresh your memories of issues that I'm sure you've come on in your previous chemistry classes, but which may have not been as salient at the time. They may have gotten foggy since then. In general, it's the availability of free energy that will drive a chemical reaction or a chemical transformation to go forward. And in biochemical systems, we're especially interested in a form of energy called Gibbs free energy, which has a combination of two components to it, of H, which stands for enthalpy, or the heat content of a system, and the entropy of it, which is S times the temperature of the system. Now, biochemical systems essentially work at constant temperature, so we can't drive a reaction by heating it up or cooling it down significantly. But these quantities, the enthalpy and the entropy, are very difficult to measure per se. And so more generally what we talk about is the change in Gibbs free energy. You may remember that when we talk about the change of a quantity, we use this capital D from the Greek alphabet. So the change in Gibbs free energy is the sum of the change in the enthalpy minus the change in the entropy, the disorder. And the guiding rule here is that a reaction will go spontaneously in that direction for the direction in which the change in free energy is less than zero, where the products have less free energy content. And one of the classical examinations of, or examples rather, of entropy, what it looks like is the difference between a billiards table like this, and then you hit it, and they're all over the place. Because there are so many different ways in which they can be all over the place, that's a higher entropy state. There's less free energy stored in that situation. Now, if that were the only time that a reaction went forward, life would be pretty complicated. But fortunately, if you can couple two reactions, if you can couple a spontaneous reaction to a non-spontaneous reaction, you can drive a reaction that would otherwise not occur by itself. And this is just the classical general principle of it. If A going to B has a free energy change, Gibbs free energy change of minus 20 kilojoules, say, and the product B going to C is not favored, it has here a positive Gibbs free energy change. If you look at them overall, if you add the two reactions together, and you add their free energy changes, the overall free energy change is minus 5. So overall, the reaction from A to C is spontaneous. And this happy property that you can add free energies of reactions that are coupled is the driving force behind all of metabolism. And so we can broadly categorize metabolism into the sort of the feeding in parts in which we want to reduce compounds. And that's not a very favorable reaction. We have to draw energy for that from somewhere. And light energy is the originating force for almost all of these reactions. And if we put in energy drawing ultimately from the sun, we can take this oxidized carbon of carbon dioxide, reduce it down. So here we have a carbon that's part of a monosaccharide, a single sugar, and it's a partially reduced carbon here. Now that it's been reduced, it has a higher free energy intrinsically and can release that through oxidation. And that release of free energy can then be used to drive some other reaction as we burn it back to carbon dioxide. So this little schematic sequence here is going to underlie a surprisingly large fraction of catabolism. Just trying to give you a look ahead so you know what to anticipate down the road. The computer changed my font again. Oh well. Always interesting to see what fonts they change. Earlier in the handout and in that chapter, there was a reminder, a review of some of the functional groups that you'll encounter many times during the course. And I encouraged you at that time to refresh your memory of all those functional groups. This table is one that shows you the various oxidation states of carbon. And again, this is, one of the ones you really want to review. And what they've done here is to line up from the top to the bottom the most oxidized form of carbon, which is carbon dioxide, in which every bond of the carbon is attached to an oxygen, down to a fully reduced carbon in which every bond of the carbon is attached to a hydrogen. Oxidation and reduction is gaining or losing electrons, but in the case of carbon chemistry, it's a matter of an unequally shared electron. In carbon dioxide, the oxygen is getting the lion's share of that electron, so the carbon is relatively oxidized. In methane, the carbon is getting the lion's share of it, so it's relatively reduced. Remember, gaining electrons is reduction. And then what you see from top to bottom is simply how many of the bonds of that carbon are to things between oxygen and hydrogen. So here all the bonds are to oxygen, here about half to oxygen, half to hydrogen, and so on. So this is the most oxidized state for carbon. It has the least free energy. Here is the most reduced form. It has the greatest free energy available to it. And so it's the one that's going to be able to provide the most energy if it's metabolized and oxidized by an organism. And again, refresh these bonds in your mind. I know you've encountered them in organic chemistry and elsewhere. We're going to come back to them. So how did all this get started? Where does this complicated self-feeding cycle begin? And what we see today are cells that make more cells. The question of where that first cell came from is one of obviously lively interest. So that one of the earliest hypotheses for this is the idea of prebiotic synthesis, that the compounds needed for it could be made without living things. And the setup that was set up to try to kind of mimic this was to have a mixture of gases in which electrodes simulated lightning, putting energy into the system from mimicking the lightning, which would have been a component of the early Earth. And then by running it through sort of in a cycle, you could have this accumulate over time and sample it at the bottom. The earliest models for this used a mixture of gases that was somewhat reducing in its own right. And we think now, for geological reasons, that probably was not as highly a reducing atmosphere as that. But nonetheless, a variety of compounds can be made this way in the total absence of life. And we can be somewhat confident that that variety is not entirely misleading, because a similar variety of organic compounds, some of them fairly complex, can be found in space and on meteorites in the absence of living organisms. One of the places that is especially intriguing to people who are thinking about the origins of life are the hydrothermal vents. Hydrothermal vents are places under the ocean floor where contact with hot magma is heating groundwater or ocean floor water and spewing up hot, sometimes very hot water out of these cracks, these vents. And that location provides a number of things at once. The heat provides simply chemical energy to put into the system. Many of times the chemistry of that vent is such that there's an availability of reduced compounds. Because you have the ocean on top of it, you have a colder area nearby that can cool this and, in fact, capture things once they've formed. And in the region around hydrothermal vents, it's quite clear that we can find things like acetic acid, even like short polypeptides that can form spontaneously. Another thing about the hydrothermal vents that is intriguing and somewhat promising as one possible location in which some of this went on is the availability of clay-like surfaces. And the clays have, it's kind of interesting, made him out of the earth is sort of an interesting metaphor. It turns out that clay is actually a good thing to perhaps start life forms from. Clays have a charged surface, a positively charged surface, and it provides a template, a very loose, foggy template against which charged monomers can line up, and the higher concentration of the monomers there then favors the formation of polymers. Another thing that makes these vents interesting places to think about as possible sites is that at the vents, some very old life forms are still present. Organisms that can literally eat rocks. They don't depend on sunlight in any sense at all. They're purely chemical eating things. Life forms that don't use oxygen at all. So they are clearly a location at which organisms that have been around for a very long time are happy, and perhaps that reflects the fact that they were initially formed there. I need to find out what font that is that got changed around again. One of the more recent hypotheses that further amplifies and builds on these, is not instead of them, but sort of builds on them, is what's been called soup in a sandwich, which is the idea that in mineral formations where sheets of the mineral can form like this, that they provide a small area in between where that soupy kind of chemistry can go on, but the sheets, the sandwich part of it, can protect it from dilution and so on. So I think it's an area of ongoing investigation, and I'm sure it will continue to be one of intrigue. In order to have something that we can recognize as life, there has to be a way to have more of it, and have more of it in a way that reflects what it came from, or what we call replication or reproduction. Replication is the essential characteristic of a living system. They're giving here an example of, just in very general terms, what an RNA replication pathway would need. They call it a mechanism. It's not a mechanism in the sense that biochemists use that word, but just the idea that if you have an original string of bases, of polymer, that free monomers, in this case nucleotides, can associate in a meaningful way against it and form a new polymer. If those chains separate, the complementary nucleotides can then make a new chain here. So when you started off with a single polymer here, you now have two that have the same composition, the same sequence. And that's the kind of thing that has to happen for there to be a living system. In recent years, we have found that there are inorganic, non-biological compounds called midwife molecules that can fit in between the bases of RNA or DNA. And these midwife molecules facilitate, can actually help with replication without having any enzymes. So the existence of these midwife molecules is an intriguing kind of whisper of perhaps the earliest steps of replication. There are also compounds that are found in meteorites and places like that, so they're not ones that are byproducts of living systems. Another difficulty of this early hypothesis that RNA was somehow the first replicating molecule is that if any of you have used RNA in a laboratory, you know that RNA is finicky. It likes to fall apart. And when I first heard RNA life proposal, I thought, but it falls apart when you sneer at it. But it turns out that one mineral in particular, which is borate, stabilizes RNA to a remarkable degree. And as a further interesting coincidence, and oh, isn't that odd, borate deposits tend to be found near the clay deposits. So there's a sort of a further hint that there's stuff around that might make that work. So if we look back to the early cells that we can actually find traces of and find minerals, fossils of, or chemical evidence for, the earliest cells had to be able to fix carbon. Fixing carbon means taking carbon dioxide out of the atmosphere or a few other sources and turning it into molecules that you actually use for living. That is clearly an essential trait of all the early cells. Very, very soon after that, a further invention happened of photosynthesis, of being able to provide the energy for carbon fixation by capturing light energy into a usable chemical form. And one of the earliest photosynthetic organisms, although not quite the earliest, are the blue-green algae. I still call them blue-green algae. You may also have heard of them as cyanobacteria. Cyanobacteria are prokaryotes, and we're going to get into what prokaryotes versus eukaryotes are. But the blue-green algae, or cyanobacteria, are especially important because they are capable of the full complex photosynthesis that the land plants can do. They can produce oxygen. And the fact that they produce oxygen literally transformed this planet. The earliest fossil traces that we have of living things go back to about 3.5, 3.7 billion years ago. Deposits of blue-green algae in big shoreline deposits that are very similar to the ones that are found today in comparable geological situations. So they've been around a really long time. That whole group of prokaryotes includes two large subgroups, if you like. There's the eubacteria that are similar to the kinds of bacteria like E. coli and so on. And then another group that has been called archaea. They were called archaea when people first started to realize how different they were from some of the other groups. They made methane, for instance. It's possible that they're not actually older than eubacteria, but they are clearly somewhat different in their biochemistry. Common characteristics of prokaryotes. And I want you to notice here that I am not repeating the list in the book. There are some differences between what I'm going to say now and the book. And so when I disagree with the book, I'm right unless you show me something else, okay? I'm always happy to learn, but I will only contradict the book if I think I've got specific evidence. So prokaryotes will have either no internal membranes at all, or they will have single membranes. They'll have a compartment with a single membrane around it. So for instance, blue-green algae have a single membrane compartment around their photosynthetic apparatus. Most prokaryotes, although not all, have some kind of wall. The chemistry of that wall is different from land plants. Prokaryotes have a nucleoid with circular DNA, almost always. So they have their genetic material organized as a complete covalent circle. We'll see why that's different from eukaryotes in a second. They are usually small. They are usually less than a micron. But there are certainly some significant exceptions, and we'll touch on that. One of the hallmarks of the prokaryotes is complicated biochemistry. The biochemistry among the prokaryotes is much more diverse than what we can do. You can find prokaryotes that will eat uranium. You can find prokaryotes that will break down testosterone. You can find prokaryotes that can do all kinds of truly strange biochemical tricks. So the great strength of prokaryotes is they're small, but they're cunning. They can do really complicated biochemistry. Because they are small and robust, and sort of many of them are pretty independent, prokaryotes are found at the extreme limits to life. You find them living in acid. You find them living in boiling water. You find them living in 5 molar salt. You know, they can live under truly extraordinary circumstances. It's worth noticing that if you consider all prokaryotes, and blue-green algae are definitely prokaryotes, but this is also true of actinomycetes and some other groups like that. Prokaryotes can be multicellular. There are prokaryotes that have multicellular organization. There are prokaryotes that have more than one cell type. So if you think of being multicellular or differentiation as hallmarks of the eukaryotes, that is incorrect. And in fact, prokaryotes can even have what are called terminally differentiated cells, cells that have acquired a subset of biochemical properties and will never grow again, like our nerves or our muscle cells. So this capacity to differentiate is not unique to eukaryotes. Prokaryotes, some prokaryotes can do it as well. Blue-green algae can have several cell types, and the cells that fix nitrogen in blue-green algae are terminally differentiated and will never grow again. So some of the things that you may have picked up as being characteristics of eukaryotes, in fact, some smart prokaryotes can pull off. Any questions about this before I go on? Okay, great. I think it can also be helpful to try to have a sense of scale. So what this picture is doing is trying to give you a little bit of a sense of that. So here you have virus particles. This is a 300 nanometer bar. Electron micrograph. Here's another, this case, a scanning electron micrograph. This bar is a little bit smaller. You can see these are definitely much bigger than those viruses. And these two scale bars are fairly similar, and this scanning micrograph is of a red blood cell. A red blood cell is a eukaryotic cell in the sense that it's in a eukaryote, but it's a pretty simple cell. Many eukaryotic cells are a good deal bigger than a red blood cell, so I think they picked it because it fits nicely in the picture. But it certainly gives you a sense that even a fairly simple eukaryotic cell is bigger than this particular prokaryotic cell. So eukaryotes. So I talked about prokaryotes. One great big division of all of living things. Eukaryotes. Eukaryotes all have internal membranes in some form or another around, in most cases, a variety of organelles. So an organelle is a little package within the cell. It is to the cell like our organs are to our bodies. Some of those organelles have 2 membranes completely around them. So a mitochondrion has 2 membranes. A chloroplast has 2 membranes around it. The hallmark of a eukaryote is a nucleus. If you haven't got a nucleus, you can't be a eukaryote. So the nucleus has a double membrane around the genetic material with pores that allow communication from the inside to the outside. And inside that compartment, there is no protein synthesis. The only things going on inside a nucleus of a eukaryote is DNA synthesis, RNA processing, but you don't have protein synthesis going on or beta oxidation or the rest of the glorious things we'll talk about. Another hallmark for the eukaryotes is linear chromosomes. Somehow or other, and there's a very cute trick that we'll learn later in the semester, the chromosomes of eukaryotes are straight pieces of DNA, not the closed circles of the prokaryotes. And it turns out to take a lot of organization and architecture to pull that off. Eukaryotes may or may not have a wall. I would say, actually, usually they do because most eukaryotes are plants, if you think about it. But if they have a wall, eukaryotes will typically have a cellulose wall. There's a very wide range of sizes. The range of sizes for eukaryotic cells is greater than the range for prokaryotes. And the great gift of eukaryotes is complicated morphology. We talked about the complicated biochemistry of the prokaryotes eukaryotes can come up with complicated morphology. All you have to do is look in the mirror, and you can see that. Because of their size and because of their complicated morphology, eukaryotes are more limited in what circumstances they can deal with. And so you do not find prokaryotes in those upper and lower extremes, the very hot, the very cold, the very acid, the very basic, and so on. And this picture here is to show you a scale here. This is a paramecium, and this is this prokaryote from the previous picture to give you a sense of relative scale. And you can see that the prokaryote in this case is really fairly comparable in size to some of the organelles of the eukaryote. So quite a difference in scale between the size of those cells. And I put together this graph because I think it helps get away from the idea that prokaryotic cells are one size and eukaryotic cells are another size. It's a spectrum, and it's a matter of where you lie on a spectrum. So here is the prokaryotic cells. They certainly cluster in here, with around the 1 micron area, but you certainly find them up as big as 10 microns. And you do find some as big as these upper ends of it, of 100 microns or even a millimeter across. Eukaryotic cells, a much larger spectrum of sizes. You notice here this is a logarithmic scale. This is in orders of powers of 10. And the very smallest of the eukaryotic cells is smaller than some of the prokaryotic cells. So it's by no means big eukaryote, small prokaryote. But the upper end of the eukaryotic cells is very long indeed. So if you think, for instance, about the length of neurons in a whale, you're talking about meters long, a cell that's meters long. Here's an example of the size of multicelled eukaryotes, and you can see that they, again, cover a large spectrum. Here we're talking about things that are actually like a kilometer across when you start thinking about some of the clonal fungi or aspens. And the mammals have a considerable spectrum, but not as large. This is a table giving you just some examples of those things. Is that showing at all? That shows actually pretty well up there. So your smallest prokaryote is 0.6 microns that I was able to find. And although 0.6 microns doesn't sound a lot smaller than 1 micron, if you do the math, you find it's actually quite a bit smaller in terms of the volume it encompasses. The smallest algae, the smallest eukaryote, is only 0.9 microns. So if you've had fixed in your head that prokaryotes are a micron across, here is an alga that is smaller than that. And I want to just point it out because they didn't even realize it was there because they weren't looking right. If you believe that all eukaryotes are at least 3 microns across, what you do is use nets that have 3 micron mesh. And then they started using smaller meshes, and they found smaller cells. This is giving you a name for that extremely big one called Thiomargarita, a sulfur bacterium. It's a clear millimeter across. You can see that cell with your eye. And this is giving you some examples of those larger eukaryotic cells. These tiny rotifers can be like 0.5 millimeter across, so they're actually smaller than a single cell of that prokaryote. This upper end here can be 5 kilometers across and be an organism. So. Here's a comparison for you. Again, a sense of scale for you. This is a transmission electron micrograph of a blue-green alga that's dividing. You can see the interior membranes. You see in here the membranes. These are photosynthetic membranes that it's going to use for that oxygen-producing photosynthesis. So clearly a prokaryote, but a prokaryote with membranes inside. It also happens to have a small cell wall, but that's not as relevant right now. And here is Ostriococcus, that smallest eukaryote I told you about. It is smaller than the blue-green alga. And what's kind of cute, if you go looking at it, it has the nucleus. It has 1 chromosome. It has 1 ER. It has 1 Golgi. It has 1 mitochondrion and 1 chloroplast. So it's got 1 of each. This is another one I thought might entertain you. These are not quite to scale. They're fairly close to scale. The gromia here is a marine amoeba, and I have on the icon a couple of supplementary readings. There are a number of things. So you bring to this class a very wide background, and some of you I know are hearing things you've heard before. So I will include supplementary readings on icon if you want to go beyond the textbook. And two of the ones I put up today have to do with with these questions of size. And one is this amoeba. Bromia is an amoeba big enough to see with your eye. And they found it because it leaves tracks on the ocean floor. They now think that organisms like it are responsible for some of the Precambrian tracks that we thought showed animals. And this is a teeny snake that was recently discovered. So I think this pair of pictures maybe says more vividly than any of my graphs can just how up or down the scale organism size can go. So, any questions about that before we move on? All right. We want to look about the evolution of cells. Mentioning evolution, to my eye, biochemistry really needs to incorporate evolution at many points, and I will do that. Some people are uncomfortable about evolution. If you have any questions or concerns, I really do encourage you to come to my office hours. We can have a chat about it. And I won't go into more detail at this point about that, but I would really encourage you to come if you have concerns. When we try to think about evolution, we have a number of kinds of clues we work with. There are biochemical ones, what kinds of pathways are present, what kind of proteins are present, and we have morphological clues, the shapes of things. So biochemical clues are evidence here in the present of the actual surviving organisms. Morphological clues may be in them or in the fossil record. And when we look at that, we discover that the eukaryotes actually have some characteristics from both groups of the prokaryotes, both the eubacteria and the archaea. And then within the eukaryotes, we have the diversity of things like animals, plants, fungi, protists, various branches that are different shapes. Looking at that contrast between the prokaryotes and the eukaryotes, you might ask, where did the eukaryotic cell come from? And there are some similarities between chloroplasts and blue-green algae, for example. And there are some similarities between mitochondria and bacteria. On the other hand, eukaryotes have a lot of compartments that don't have any genes at all. Mitochondria and chloroplasts, both of them have some genes, not enough genes, but some essential genes for making themselves work. But there are a lot of other compartments that are entirely coded in the nucleus. And so this gave rise to a theory for the origins of eukaryotic cells where one cell or several cells sort of incorporated other cells into it. And the idea here is that maybe this is the blue-green alga and this is the proto-mitochondrion, and by one of them eating the other, you end up with the earliest eukaryotic cell where you've cannibalized or parasitized these other organisms. One of my difficulties with this theory for where these organelles came from is that it suggests that the anaerobic precursor was able to eat these other cell types. In the world that we know, anaerobic eukaryotes are not capable of eating things. Eating things is a very energy-expensive process. I'll just mention without, I'll show one other diagram for it, that an alternative to the origins of the eukaryotic cell is the idea that some prokaryote that had these capacities compartmented, and then maybe there's gene transfer between the compartments. This is a figure from a discussion of where the nucleus of the eukaryote came from. Obviously, any origin of the eukaryotic cell has to account for the nucleus in the eukaryotic cell. And this is showing you one proposal for how that might have happened, that if you had DNA stuck to the membrane, as it is in prokaryotes in most cases, and membrane-bound ribosomes, that if the membranes of the cell periphery kind of came around and kind of enclosed that DNA, you start to have a structure called a mesosome. And if you continue that extension and elaboration of the membranes, you could, in concept, in principle, hypothetically, enclose those membrane-bound DNAs completely so that they now are in a compartment. And this business of the membrane-bound ribosomes then becomes part of the ER that's kind of an extension of a nuclear envelope. So this theory for the origins can account for both the double membrane character of the boundary around the nucleus and why the endoplasmic reticulum often is attached to the nuclear envelope. And so I would just sort of point out that if this is what were the origins of the nucleus, invoking this sort of engulfment pathway for mitochondria is invoking a second pathway when we've already identified one. But it is a matter of ongoing investigation. It's not something where we have a final answer yet. Okay. I think that the rest of this handout is some of those functional groups from Table 1, and I'm not going to go through them right now, but they're there. They've been multiplied for you to look at. Do you have copies of Chapter 2? Is there anybody who does not have a copy of the handout for Chapter 2? Could you help there? Thank you. And we will start that. Any questions about Chapter 1 before we start on Chapter 2? If you need Chapter 2, please hold your hand up high. And can you work on that then? Thanks very much. Okay. So we're going to, I think in the future we'll try to have the handouts a little further forward for you to pick up. But today we distributed Chapter 2 because, as you can see, we're going to start it before Wednesday. In general, we'll try to have the handout for a chapter on the day that we start that. In most cases, that'll be the day that's actually on the calendar. Any questions about Chapter 1 while he's handing out the Chapter 2 handouts? Questions? I guess asking people to hold up a hand. Wave at me if you have a question. Okay, so I'll start working on Chapter 2 then. Okay. So when I started biochemistry as a graduate student, the professor who started the course began with, what would life be like without water? And the answer was crisp. What we're going to talk about in Chapter 2 is aqueous chemistry. Some of this is, again, review, but it's probably review from a different perspective than you've seen it in the past, or perhaps you didn't have the motivation to absorb it at the time. And part of it is going to be some novel aspects of it. Water is essential for life for a number of different reasons. Number one, the kind of solvent that it is. And we're going to look at why water is such an amazing solvent. Secondly, the properties of water itself are what drive the formation of structures of proteins and membranes. And we'll take a look at how and why that's true. And in addition, acid-base relationships are going to be an important part of a lot of the enzyme mechanisms and how things happen in living systems. And so we're going to look at that a little bit as well. Because life is tied to water, because water can do such essential things, it turns out that that itself is part of what sets the limits for living systems. So in order to have liquid water, you've got to be between 0 and 100 Celsius. So that becomes, almost by definition, the parameters that living things work in. It also means that that's the sort of the parameters that limit the chemistry of living systems. That if it's not compatible with exposure to water, living systems are not going to probably take advantage of it. Now, what happened to my fonts? Okay, this is back. All right. Interestingly, we are substantially made out of water. We're over 60% made out of water. If you look at cells, individual cells, you can find cells typically having 50% water and 50% other stuff. What's striking about that is that means that a cell is as densely packed as a protein crystal. If your usual notion of a cell is kind of like an empty balloon with things kind of floating around, it's much more busy. It's much more like Times Square on New Year's Eve. And where's all that stuff hiding out? I need to fix my pointer. Let me get that visible. And so what do we have there? We've got intracellular water is about 40% of our mass. Interstitial water is water in the little spaces between cells and between tissues and stuff. The circulatory system, which is probably where you think of the water being, in your bloodstream and so on, is about 5%, and then about 40% other stuff. You may remember that on Friday I pointed out to you HIP casino, the 6 most abundant atoms and their electronegativity scale. You know, with HIP being hydrogen being the least electronegative and casino, oxygen, being the most electronegative. Well, water is made out of hydrogen and oxygen, the least and the most electronegative atoms. And oxygen allows for a very interesting structure to the molecule overall. So the oxygen atom is here at the center. Here's one of the hydrogens. Here's another hydrogen. And there are these 2 unshared electron pairs kind of sticking out of the backside of the oxygen. So it's this tetrahedral molecule. Because of the very big differences in electronegativity, The electrons that make up the bond between hydrogen and oxygen are not shared equally. And as a consequence, that oxygen atom has a partial negative charge. It's not a full negative charge, it's a partial negative charge. And the hydrogen atoms have a partial positive charge. It's not a full positive charge, or it wouldn't be part of the molecule, but there is an unequal distribution of electrical charge across the molecule. Therefore, water is a polar compound. Both the bonds are polar and the molecule itself is polar, as we're going to see in a bit. Because the hydrogens have a partial positive charge, they're capable of forming hydrogen bonds. And if you look at this, you can form up to 4 of them. These are the hydrogens that can participate in hydrogen bonds as partial positive charges. And then the back side is where the partial negative charge is visible, so to speak, and it can form hydrogen bonds off the back side, so to speak. So it can form up to 4 hydrogen bonds on any one water molecule. And this is just showing you those partial charges with the Greek deltas. And what you can see is that because the hydrogens are sort of, up there like that, goalpost position, the molecule as a whole is polar. That there's sort of a positive side to it and a negative side to it. And that has further implications. But this partial charge character over it means that it can interact electrostatically with a very wide variety of molecules. So this is showing you diagrammatically a hydrogen bond between the partial negative charge on this oxygen. Now we're using a ball and stick representation. Remember, we talked about different representations for molecules. So this is the ball and stick with its partial negative there and the partial positive. You can see that there's an alignment of that hydrogen. So the better the alignment is of the bond of that hydrogen to the oxygen toward the recipient, the stronger that'll be. And in some cases, the particular positions of the donors and recipients is such that they can be aligned very well, and instead of being more with this oxygen than that one, this hydrogen is halfway between, in which case the hydrogen bond is a bit shorter and a bit stronger. And that's called a low barrier hydrogen bond. Now because of, what is with these fonts? Excuse me. Now because of this capacity to form multiple hydrogen bonds, liquid water and ice have really remarkable structures. In ice solid, each water molecule completes all 4 potential or possible hydrogen bonds. So every single potential bond is an actual bond. And this is a diagram over here of a water crystal. It is a crystal, although we may not think of it that way. And because of that spacing of things off in 4 directions, the overall structure of ice is less dense. It's more distributed and diffuse than liquid water. This is very unusual, very unusual. To have the solid be less dense than the liquid is quite remarkable. In water, any one water molecule may have up to 4 hydrogen bonds, but those hydrogen bonds are transient. They're being formed and broken all the time. But the abundance of those bonds makes water very cohesive as a fluid, and cohesive in a way that, good Lord, I need to find out what's going on there. I don't know why my fonts are changing, because this was not partly hidden when I made it. And you can see here how any one of these water molecules has several bonds, but they don't all have 4 bonds like the ice. So this polar character and this capacity of making so many hydrogen bonds per molecule gives water a very unusual set of characteristics. It has a high surface tension. And so that's sort of diagrammatically shown here, this bug that's literally walking on the surface of water because the surface tension where the water encounters the air has a coherence to it that most liquids would not have. It has a high melting point for its mass. It has a high boiling point for its mass. It has a high heat capacity. This is a great thing. It means that living systems are embedded in a medium that's good at absorbing heat and buffering the heat things. The polar character of it makes it a good solvent, and we'll look at that in a little bit more detail. And the fact that the ice is less dense than the water makes life possible on Earth. Think about it for a moment. Suppose ice were more dense than water. What would happen? Every time you get some icebergs forming on the pole, near the Arctic or something, it would sink to the bottom where it's cold, and more ice would form and it would sink to the bottom where it's cold, and you'd end up freezing all the ice and getting it out of the way. You'd end up with a planet full of ice. And so the fact that ice can float is also one of its characteristics that makes life possible. Okay. This is irritating because this arrow is not in the right spot because the font is different. This should be pointing to the hydrogen. Sorry, not the oxygen, not the ionic. Every once in a while, Microsoft changes its system, and when it changes its system, things change that I don't know about. All right. This arrow should be coming off of hydrogen, all right, not off of ionic. One of the characteristics of bonds is their length, how close or how far apart the two atoms that are being bonded are, and then their strength. And different bonds are going to have different lengths and different strengths. We can, generally speaking, about four classes of bonds we're going to encounter in living systems. These hydrogen bonds that we've talked about that are not terribly strong hydrogen bonds. Ionic interactions, as you can imagine, are between a full positive charge and a full negative charge. Or an actual covalent bond where those electrons are shared and make a covalent bond. The weakest of all interactions is van der Waals interactions, which are the ones we think of that sort of keep atoms from being embedded in each other. They're just sort of at the surfaces of things. And it's because these hydrogen bonds are kind of intermediate in strength between all of these that they end up being so powerful when you take them all together. Hydrogen bonds can be found in living systems with oxygen as they are with water. Hydrogen bonds can form with nitrogen and with sulfur. So there are a number of places where we can see them turning up. I like this diagram that they have for sort of the powers of the weak. This is taken from Gulliver's Travels. If you know Gulliver's Travels, Gulliver found a world where everybody was this tall. And you'd think, well, you'd be in charge of everything if you're that big. And this is the diagram showing you how Gulliver was captured by the Lilliputians. Because although no one of them was very strong, when 1,000 of them started to tie down everything they could reach, he couldn't overcome it. And it's those multiple weak interactions that makes life possible. This is showing you diagrammatic the length of these different bonds that are typical, not all, but a typical covalent bond might be an angstrom long. Here's a hydrogen bond, 1.8 angstroms. And then this is the distance that's sort of set before you actually start to have van der Waals bonds. We're going to see other kinds of electrostatic interactions. So a hydrogen bond is one kind of an electrostatic interaction. We're going to see other ones as well. Here we're seeing base pairing between cytosine and guanine. This is an example of electrostatic interaction from a carbonyl oxygen to the backside of a carbon. These are hydrogen bonds in here. London dispersion forces are ones where fluctuations in the distributions of the electrons generate a local charge for a moment, and it induces a local charge on the other side. They're very weak kinds of interaction. Let's look for a little bit at water as a solvent. I am so ticked about these fonts. I'm going to have to work this out. So it's a really extraordinary, we've talked about this capacity for hydrogen bonding, and what it means is that water can do an extraordinarily good job at hydrating or solvating. So hydrating because it's coating with water, solvating because it's coating it as a solvent. Both ionic compounds and polar compounds. And this is a really good thing because the cell is a very crowded place. This is a diagram from someone who makes a point of trying to be realistic about the space taken up by things in an actual cell. And you can see there really isn't a lot of spare space inside of cells. And what it means is that in the level of crowding that cells show, there's only one to a few water molecules deep around many of the water molecules. And so the water can act as kind of a bumper, protecting molecules from others. And we've come to 4:20, so I will say, see you on Wednesday. AV person, you can stop recording. Folks, don't forget that we've got the